Hey there, and welcome to this explainer. Today, we're taking a classic scientific tool and basically turning it into your own personal spaceship for discovering hidden, microscopic worlds. We are, of course, talking about the compound microscope. Whether you're right in the middle of grade 7 science or you just have a super curious mind, mastering this incredible device is absolutely your first step into a whole universe that's completely invisible to the naked eye.
But before we really get going, I want you to think about something. How do scientists actually study things that are totally invisible to us? I mean, things like the individual cells that make up your own body, or the tiny little organisms swimming around in a single drop of pond water. Well, the secret lies in bending light and using lenses to trick our eyes into seeing the ultra small.
So here is our flight plan for today's explainer. First, meet the microscope. Then, mechanical parts, illuminating parts, magnifying parts, and finally, making a wet mount. Section 1, meet the compound microscope. All right, so the compound optical microscope, which, by the way, just means it uses light to enlarge images, is cleverly split up into 3 logical groups. You've got the illuminating parts that provide the light, the magnifying parts zoom in, and the mechanical parts that hold the whole shebang together. Section 2, mechanical parts, the foundation. Let's kick things off with a key structural component, the arm. You can really think of these mechanical parts as the sturdy chassis of our scientific vehicle, right? The arm is that curved support connecting the upper optical parts to the base. And the absolute most crucial point here is safety and stability. Whenever you're carrying your microscope across the classroom, you've got to always use one hand to firmly grip that arm and place your other hand totally flat under the heavy base. Next up, we have the stage. This is literally where the magic happens. It's the flat platform where you securely place your specimen slide. It uses these little metal pieces called stage clips, and they lock your slide into place, right over a small opening. Now, that opening is super important because it allows light from below to pass straight up through your specimen. Once your slide is on the stage, you're going to need to focus it. You have 2 main tools for this on the side of the device. The coarse adjustment knob is the big one. It moves the stage up and down pretty rapidly for your initial rough focusing. But be careful here. Once you get close to being in focus, or especially if you switch to a higher magnification, you absolutely must switch over to the fine adjustment knob. This one makes tiny, precise movements to give you a sharp, crystal clear image. Section 3, illuminating parts, lighting up. Down right at the base of the microscope, you'll find the illuminator. You can think of these illuminating components as the headlights of our microscope. The illuminator provides a nice, steady, built-in light source. Alternatively, some models use a mirror to actually catch external room light and bounce it directly upward. But either way, this light is shooting straight up through the bottom of the stage to hit your specimen. Above that light source is the diaphragm, specifically called the iris diaphragm. Now think of this as acting exactly like the pupil of your own eye. Sitting just under the stage, it uses these adjustable, overlapping blades to control the exact amount of light that reaches your specimen. And why does that matter? Because opening or closing this diaphragm optimizes your contrast, which lets you see highly detailed, transparent structures way more clearly. Section 4, magnifying parts, zooming in. So, with our samples safely supported and perfectly illuminated, it's time for our zoom lenses. First in this optical system is the eyepiece, which is also known as the ocular lens. This is the lens located at the very top of the body tube. You know, the one you actually look through. Typically, the eyepiece alone provides a baseline magnification of 10 times. That means it automatically makes whatever you're looking at appear 10 times larger. Now, moving down the microscope, we hit the revolving nose piece. This is a rotating turret located just above the stage. It lets you easily switch between different levels of magnification simply by turning it. And honestly, you'll hear and feel this really satisfying click when a new objective lens perfectly locks into place right over your specimen. The actual lenses attached to that nosepiece are called the objective lenses. A standard lab microscope usually has 3 or 4 of these, and they're usually color-coded. You'll typically see a 4x scanning objective, a 10x low power, a 40x high power, and sometimes even a 100x oil immersion lens. Now, here's a quick trick for calculating your total magnification. You just multiply the objective lens by the eyepiece. So using a 40x objective with your 10x eyepiece means you're seeing that specimen at an amazing 400 times its actual size. No way, right? Section 5, making a wet mount slide. Okay, so you're totally confident with the microscope's parts now. Let's put that knowledge into action.
Sometimes we don't have nicely prepared slides ready to go, so we have to make our own fresh slides right in the lab. For this practical application, we're going to act like real biologists and make a wet mount using our very own human cheek cells. It is just a fantastic, hands-on way to put your new microscope skills to the ultimate test. First things first, gather up your lab materials. You're going to need a standard, clean glass microscope slide and a tiny, very delicate square of glass called a cover slip. You'll also need a clean toothpick, a small piece of paper towel, and a special chemical stain called methylene blue. Let's walk through the exact procedure here. Step 1, take that clean toothpick and gently, but vigorously enough, scrape the inside of your cheek. Step 2, smear the end of the toothpick right in the middle of your clean slide. Step 3, carefully add exactly 1 drop of methylene blue right onto that smear. And finally, step 4, gently place the cover slip over the droplet using your paper towel to just soak up any excess stain that might escape out the edges. Now, you might be wondering, what's up with that methylene blue? Why do we need it? Well, many cells, like your cheek cells, are naturally almost completely colorless and transparent. Staining is simply a process that uses a chemical to alter the color of a specimen. It's an absolutely vital step because it creates contrast, making those hidden internal components of the cell visibly pop right out against the bright background created by your illuminator. Thinking about the entire compound microscope as a whole system one more time, the absolute key takeaway here is synergy. By understanding exactly how the mechanical foundation, the illuminating headlights, and the magnifying zoom lenses all collaborate together, you are now fully equipped to operate this amazing tool safely and effectively. So, as you head into your next grade 7 science lab session to finally see your own cheek cells up close, I want to leave you with this final thought. Armed with your compound microscope, what other invisible mysteries of the natural world are you most excited to uncover next?